In this video, we're going to talk about a parasite that causes amoebic dysentery or a disease known as amoebiasis, and the name of the parasite is known as Entamoeba histolitica. Now, in the last video, we talked about parasitic amoeba, and we were able to mention that we have four genera of parasitic of amoeba that causes disease known as amoebiasis, and we mentioned them to be Entamoeba, iodomoeba, endolimnax, and dientamoeba. And we say most of these are commensals, even though they are parasites, but they are non-pathogenic parasites. We call them commensals because they do not really cause disease. But the major parasitic amoeba that causes disease known as amoebiasis is Entamoeba histolitica. So today we will be talking about the biology of Entamoeba histolitica. Entamoeba histolitica is an eccentric protozoan, parasite of man and other mammalian hosts such as dog, cats, and other primates, and it causes a disease known as amoebic diarrhea or amoebic dysentery, also known as amoebiasis. It is estimated that about 50 million people from tropical and subtropical region is infected with this particular parasite. And so I said about 50 million inhabitants of tropical and subtropical region in subtropical and subtropical countries are infected with this particular parasite, and number of children in developing countries are at risk of this particular parasite. So let us check the systematic position of this animal. Let us check the systematic position of this animal. Now look at the systematic position. They are in kingdom Protista, the subkingdom Protozoan, the phylum Sarcomastigophora. They are in subphylum Sarcodina, superclass Rhizopoda, class Lobosae, order Eudomoeba, genus Entamoeba, and species Histolitica. So this is the what? This is the systematic position, or let me say the classification of Entamoeba histolitica. Now, what is the geographic distribution of this animal? They are found in all populations throughout the world. Hence, I can say they are what? They are cosmopolitan in distribution, and it is more prevalent in tropical and subtropical region rather than cooler climates. So they are not really found in a place where their weather is actually cool, but they are found in what? They are found in tropical and subtropical what? Tropical and subtropical region. Now, what about their habits and habitats? What about their habitats? Trophozoite of Entamoeba lives in the mucus and the submucous layer of the large intestine. Hence, I can mention in my own word that Entamoeba are endoparasites that lives in the upper part of the large intestine, that is the colon of a man, and they feed mainly on tissue of the intestinal wall and often produces severe ulcer and abscesses. You understand? In chronic cases, they may enter the blood circulation to reach the liver, the lungs, the brain, and even other organs. And the cause is very serious disease known as amoebic dysentery or amoebiasis. So this is their morphology. This is the morphology. The size is about 12 to 60 micrometer. The ratio of nuclear material to cytoplasm is 1:10 to 1:12. The nuclear morphology is the nucleus is round, vesicular with central choriosome and peripheral chromatin. The ingested material, they ingested RBC, that is what they feed on. They feed on red blood cell, and the cytoplasm is uniform and finely granular, and the trichosome staining, we have green cytoplasm and dark red nucleus. So that the reason why they have dark red nucleus, you understand. Now let's talk about the forms that it takes, their morphology. They have two morphological states. You understand? They have two morphological states. But in this material, I will say three morphological states. You understand? The two morphological states is the trophozoite stage and the cystic stage. You understand? So under the cystic stage, we still having pre-cyst stage and the cystic stage. You understand? Now, the trophozoite is known as the active or diagnostic stage of Entamoeba histolitica. The active or what? The active or diagnostic what? Stage of Entamoeba histolitica. While the cyst stage is the dormant stage or the dormant stage, the dormant stage of Entamoeba histolitica. So that is what the cystic stage is. Now let us talk about the characteristic of the trophozoite stage. The characteristic of the trophozoite stage. Now, under the microscope, the living parasites on worm stage exhibit a remarkable locomotion. It exhibits a slow gliding movement using their what? Using their pseudopod. Remember that it is pseudopodia that they are what they are using. Now look at it. This is the structure of what? This is the structure of Entamoeba histolytica trophozoite. The trophozoite of Entamoeba histolytica. So this is the what? This is the pseudopodium. Can you see? This is the pseudopodium of Entamoeba histolytica. And this is the what? This is the endoplasm. This is the karyosome. This is the nuclear membrane. The ingested red blood cell. Remember I told you that they ingest red blood cell. And this is the what? This is the ectoplasm. So they have two. They have the ectoplasm and the endoplasm. And both the ectoplasm and the endoplasm forms the what? They forms the cytoplasm. You understand? Now, they are the vegetative or the feeding phase of an amoeba. That is the trophozoite stage. And they are amoeboid in shape. The Magna strain of this particular animal varies between 20 to 40 micrometer, and the Minota strain varies between 50 to 20 micrometer. So they have the Magna strain and they have the what? The Minota strain. That is the big strain and the small strain. They have a clear outer ectoplasm with a granular endoplasm. Now look at this. Look at their ectoplasm. It is clear. You understand? While their endoplasm is what? It is granular. Are you getting that? Now, the endoplasm consists of a blood-filled food vacuole and starch granules. Look at it. This is what? This is a food vacuole. And the food vacuole consists of what? The food vacuole consists of red blood cell. Are you getting that? So this is the food vacuole as well. Consists of red blood cell. So the ectoplasm and the endoplasm, the both of them forms the what? Forms the cytoplasm. Are you getting that? And this is the what? This is the nuclear membrane and this is the karyosome. Now, moving forward, they have a single vesicular nucleus. I said that already. That is with a centrally placed endosome. Endosome is also known as karyosome. Now look at it. Look at it here. This is the nuclear membrane. This is the nucleus. Look at it. That I marked in green. This is the nucleus. And they have a what? They have a central place what? Central place karyosome or an endosome. So their endosome is what? Is centrally placed. Are you getting me now? And this is the what? This is the characteristic of the trophozoite form of Entamoeba histolytica. Now let's talk about the cystic stage of Entamoeba histolytica. Now, like I mentioned, I said they have two forms. They have the precystic form and the cystic form. The precystic form is colorless, round, oval, and smaller than the trophozoite, but it's larger than the cyst. So it is in between, smaller than the trophozoite, but larger than the cyst. You understand? And they have a blunt pseudopodium, and the characters of the nucleus remain intact, still remain the same way as what? As the trophozoite. Still a central placed karyosome, as in trophozoite. Now let's talk about the cystic stage. The cystic stage are encountered only in the lumen of the intestine under unsuitable condition. That is where you can find the cystic form. And the cyst is round or oval in shape. Are you getting me now? They are refractile and purely white in color. And the cystic stage begins as a uninucleate body, but divide by binary fission and develop into binucleate, then to quadrinucleate structure, which we'll be talking about in their life cycle. So they have what? They have a uninucleate, then it transforms into binucleate, then into quadrinucleate. Quadrinucleate means what? It have four nucleus. So it starts with one nucleus, two nucleus, then four nucleus. You understand? So that is how the what? That's how their life cycle goes. So moving from there, the cyst is about 6 to 15 micrometer in diameter, and a distinct glycogen mass is found in the early stage of cyst formation. I get me now. The newly formed cyst consists of three things. Consists of a nucleus, a glycogen mass, and a chromatida bar, a chromatidal bars. You understand? So it consists of what? Consists of a nucleus, the glycogen mass, and a what? A chromatida bar. So look at it. Let me just illustrate that. Let me just illustrate that. Let us say this is what? This is the um this is the ectoplasm. Sorry, let me make that one to be thick a little bit. Yeah. So let me say for example, we have something like this. Let me clean this. We have something like this. This is the outer ectoplasm, right? Let me say this is the outer ectoplasm, or let me say this is the endoplasm. Then we have the what? Sorry, it is not really smooth. Then inside the endoplasm, we have a nucleus. In the cyst, we can have a quadrinucleate. Let me say, for example, this quadrinucleate, four nucleus, and we have a chromatida bar, something like this, chromatida bar. You understand? And we have a glycogen vacuole. We have a glycogen vacuole. Let me just use this one as an example, as a glycogen vacuole. You understand? So this is what? This is an example, a typical example of what? Of a cystic form of Entamoeba histolytica. Are you getting that? So that is just an illustration of that. So that is what they what? That is what they contain. That is what they are trying to like say here. A chromatida bar, a glycogen mass, and what? And a nucleus that is stained with what? Iodine. Now, let's talk about the life cycle of Entamoeba histolytica. The life cycle of Entamoeba histolytica. Now, this is the what? This is an example of this is the life cycle of Entamoeba histolytica. Now, now they have five successive stages. Five for successive stages. They have the trophozoite stage, the precyst stage, the cyst stage, the metacyst, and the metacystic trophozoites. I mention again the trophozoite, the precyst, the cyst, the metacyst, and the metacystic trophozoites. You understand? Now, the life cycle begins when an effective cyst is ingested when an infective cyst is ingested through contaminated food or water or via sexual contact in homosexual men. They can be contaminated through what? Through food, water, or sexual, like anal sex within what? Homosexual men. Now, encystation now occurs in the small intestine. Look at it. This is encystation. It occurs in the what? It occurs in the small intestine to release four four nucleated trophozoites in the terminal ileum. That is what they release. Now look at it. This is the trophozoite. They release the trophozoite. Can you see that? They release the trophozoite. Now, the nuclei divide to form 8 metacystic trophozoites. Divide to form what? 8 metacystic. They can't really show that inside this diagram. You know, they are dealing with only one entamoeba. These metacystic trophozoites now feed on bacteria, tissue debris. Are you getting me now? To develop into what? Develop into a matured trophozoite. This matured trophozoite now migrate into the colon to colonize the mucous layer. Are you getting me now? Now, usually the trophozoite remain in the lumen as a commensal, multiplying by binary fission. Now look at it. Remains in the lumen as a commensal and it's multiplied by what? By binary fission. Now look at it. Look at this is the trophozoite that invades the intestinal mucosa, the mucous wall. Do you understand? Now, as fecal matter passes through the large intestine, if the fecal matter, like fishes, is passing through the large intestine, this is the large intestine, can you see? It's passing through the large intestine. Now, dehydration occurs and the parasites are stimulated to encyst. They are stimulated because of the hydration that occurs. The parasite is stimulated to encyst. Now, once they encyst, the trophozoite is now condensed into spears, like something like a spear. Now, this spear is called the precyst. I get it now. It's called the what? It's called the precyst. Now, this precyst loses their food vacuole to form a glycogen vacuole in their cytoplasm. And this precyst secretes a thin iodine cyst wall to form a cyst. So, precyst develop to form a what? To form a cyst. I get it now. A cyst. The young cysts have only one nucleus. The young cysts have only what? Only one nucleus. And this one nucleus divides rapidly by binary fission to form two nucleus. Then after from two, then divide to form four nucleus, which is quadrinucleate cyst. Are you getting me now? Quadrinucleate cyst. Now, this quadrinucleate cyst is most common in formed stool. In like formed stool, you know, stool that is formed. Stool means fishes. You understand? In a formed stool, that is where you can find what? That's where you can find a quadrinucleate cyst. And these cysts are passed out in the fishy and transmitted into another susceptible host, maybe a child that is playing with or you eat a plant that is contaminated with what, with fecal matter that is consisting of quadrinucleate cyst, then it is now ingested by what, it is now ingested by another man, and the life cycle continues. But it should be noted, they have two form. They have the cystic form and the what, trophozoite form. The cyst form is found in a formed stool, while the trophozoite form is found in a diarrhea or unformed stool, watery stool. That's where you can find what? That is where you can find the trophozoite. So the cyst form is found in what? In the formed stool, and the trophozoite is found in what? In a diarrhea. So that is what? That is just the life cycle of what? Of Entamoeba histolytica. So this is the write-up that I actually. So this is how it looks. You can pause that or screenshot. So we have something like this. So this is the what? This is another illustration that we made. This is another illustration. I think this illustration is much more better. So ingestion of the cyst from food and water, the cyst that is not digested by the gastric juice, and the cyst digested inside the intestine by trypsin, encystation occurs, tetranucleate amoeba escapes, invasion of the mucous membrane of the intestine, flag-shaped ulceration of the intestine. So they can cause two things. Either it causes dysentery, you understand, or it spread into the liver, the lungs, and the brain to cause secondary infection. They can cause secondary infection as well. So look at this. This is the form. Look at it. This is the trophozoite form. The trophozoite form having only one nucleus, you understand, and the ingested food containing of what? Red blood cell. This is the precystic form. Can you see that it is still looking like the trophozoite form, but there is nothing like food vacuole again. They have lost their food vacuole. Do you understand? Now it's now moved from there to the uninucleate cyst having only one nucleus with chromatidal bars, what I just illustrated in the other time, with chromatidal bars. Then move from there to binucleate. Can you see they're having two nucleus, one here, the other one here. Then chromatidal bar as well. Are you seeing that? So they move from there to quadrinucleate cyst. But what you have to understand the quadrinucleate cyst is that they have four nucleus and there's nothing like chromatidal bar. So that is what? That is the biology. That is the life cycle of Entamoeba histolytica. Now we can move from there to the pathogenicity. Invasiveness depends on the particular zygodyms, and in their pathogenicity, they causes what? They causes a disease known as amoebiasis. Are you getting me now? Now, they adhere to the host cell with the aid of galactose-specific membrane lectin. Killing of the host cell by a pure-forming peptide known as amoebopure. That is what they use in what? In killing the host cell. I did not put that one here. So they kill the host cell with the aid of what? Amoebopure. Now, they undergo something known as proteolysis of the host extracellular matrix, which is mediated by cysteine proteinase. So these are the things that they causes. You understand? Now, talking about their pathogenicity, talking about their pathogenicity or pathology. Bacteria may enhance pathogenicity. You understand? Man is the reservoir of the infection. An infection occurs by quadrinucleate cysts. Entamoeba produces dysentery with frequent passing of stool mixed with mucus and blood. Intestinal lesion are acute amoebic dysentery and chronic intestinal amoebiasis. We can have extra intestinal lesion, that's metastatic. We can have liver amoebic hepatic. We can have amoebic liver abscesses. We can have lungs can infect the lungs. It can infect the brain. It can infect the spleen, and it can infect the skin that is near the visceral lesion. Now, that is the pathogenicity that we are going to talk about here. We have mainly we have three. We have the intestinal amoebiasis, we have the hepatic amoebiasis, and we have the pulmonary amoebiasis. The intestinal amoebiasis is the one that infect the intestine. The hepatic amoebiasis is the one that affect hepatic region, like livers. You understand? Liver is the hepatic organ, and the pulmonary is the one that infects the other organ, like the brain, to come to cause a cerebral abscesses, like the skin, you understand, to cause amoeboma, and this can infect the penis, you understand. It can infect the kidney, the spleen, the male and female genital and pericardium. These are the things that they can infect. Now, what are the symptoms of this particular disease. The symptoms is they have abdominal discomfort and flatulence, and also constipation, fatigue, and anorexia. These are the things that they can actually cause. So, what is the treatment? A severe amoebiasis can be treated with metronidazole. They can be treated with metronidazole because it is low in toxicity and is effective against both extraintestinal and colonic infection. Now, this is where we are going to stop in this particular video. I hope you have learned one or two things, and subscribe for more videos. Thank you.